Okay. So now in this part of the presentation, we're talking about the different metrics that were used for just protein structure prediction over many years, but that will feed into the metrics that AlphaFold and other programs put out. So the one that the CASP meetings used the most was something called GDT-TS. GDT stands for Global Distance Test. TS, I think, just was the coordinates. I don't know what TS stands for. It doesn't matter. And the way it worked, it really was, we would get models and we were comparing them to the crystal structure. Sometimes the participants wouldn't model the whole protein, they'd only model part of it. But what we want to know is, can they model the whole target, the whole experimental structure? So the way this works is you take the predicted structure and the model, and you align the predicted structure to the model. And the first time you do it such that you're maximizing the number of residue pairs, C alpha, residue 1, C alpha, residue 2, C alpha, residue 3. As many of those as possible to be less than 1 angstrom apart. So it could be that some part of the protein is really a rock and it's exactly the same between target and model, and you can model a lot of that within 1 angstrom, but maybe the other parts of the protein you can't. So you get the number of C alphas that are better than 1 angstrom. Now you align it again, but with the constraint that you get as many as possible within 2 angstroms. Then you do it again for 4 angstroms, and then you do it again for 8 angstroms. So you're getting 4 integers. Each of them is the number of residues that are within that distance, optimized to get as many as possible for that distance. So that's 4 numbers, and you simply say, well, each of those is a proportion of the experimental structure. Maybe it's 50% of the structure, it's 90% of the structure, and you just average those 4 numbers. So the GDT-TS is 100%, N1 divided by the length, plus N2 divided by the length, plus N4 divided by the length, plus N8 divided by the length, and you're just taking an average. So that's why roughly when I introduced it in the CASP slide, as roughly the percentage of the structure that is correct within different tolerances. It does sound a little contrived, but it does work just fine, and it has been very useful in this study. And also was useful in the competition, because sometimes people would only get a part of the structure right and they wouldn't get the rest of it. The one that is used in AlphaFold, so we do need to understand what LDDT is, because in AlphaFold there's something called pLDDT, which just stands for predicted LDDT. But LDDT came up long before pLDDT did. The AlphaFold people didn't invent it. And that stands for Local Distance Difference Test. So you get one of these numbers for every amino acid, and it's telling you something about the environment around the amino acid and whether that structure is predicted correctly. That's why it's called local. So we'll start with a crystal structure. It's my favorite 1OL5 Aurora kinase. And we're going to look at this residue right here. And within some sphere around that residue, I'm going to make a list of all of the atoms in the experimental structure, all of the other C alpha atoms that are within some distance, like 15 angstrom. And I have marked the distance between that one and not all of the ones that are within 15, but a whole bunch of them, 7 of them. So this one is 5.7 angstroms away. This is 6.2, 7. So there's 7 numbers there. They're all distances between my residue of interest, query residue, and the neighborhood around it. Over here, I have an AlphaFold model, predicted structure, and I can measure the same set of distances. And those distances are different. 5.7 is now 4.8, 6.2 is now 7.2. The structure of this loop is different than the structure of this loop, which means the local neighborhood is maybe not so good. And so I just write down that list of distances. Again, I have 7 distances here, 7 distances here. I calculate all the differences in those distances. Is it bigger? Is it smaller? And just the absolute value. Is it 2 angstroms bigger, 2 angstroms smaller? Then I write down 2. So for those 7 distances, I write down these numbers, 0.9, 1.0, 1.2, all the way up to 0.4, kind of in order clockwise. And then it's very much related to the GDT-TS that I just talked about. I'm now going to say, well, what percentage of those distances are better than 0.5 angstroms? What percentage of those distances are better than 1 angstrom? What percentage are better than 2? And what percentage are better than 4? So that's 4 percentages. The LDDT is just the average of those 4 percentages. So that's 1 out of 7, 3 out of 7, 4 out of 7, 7 out of 7 for 0.5, 1, 2, and 4. And if you do that, those 4 fractions divided by 4, multiplied by 100, you get 54%. Is that clear? It's an important number because and I think in AlphaFold, it's a 15 angstrom radius. It might include all of the, not just C alpha atoms, but all the other atoms as well. You can do it different ways, but it always works the same way. A very important score, and you all, if you've looked at AlphaFold, you hear about ipTM or pTM. So here's TM, just the plain TM. TM comes from Template Modeling Score, and it came out in 2004 by Yang Zhang and Jeffrey Skolnick. And again, it was one of these things for structure prediction. It was, well, if you predict a structure, how much of it did you get right? And what kind of score could we attach to that? And so again, you're doing an alignment of the two proteins. You might do many different alignments. You might use different alignment programs. You might align on different parts of it. So you've got a whole set of alignments. And from each of those, you can measure these distances. Remember, residue 1 to residue 1, 2 to 2, 3 to 3, just like we did for RMSD. So those are these numbers dj. So j going from residue 1 to the length of the target, to the length of the protein. And I'm going to look at this little function here. And for every residue, I'm going to evaluate that function, and then I'm going to take the average because it's 1 over the length of the target. But what is this function? Yeah, it's a little complicated. It's 1 over 1 plus dj divided by some number, d0, and I'm going to square that. Why? If I have a very good model of the protein that is maybe, all of the distances between 1 to 1, 2 to 2 are 0.1 angstrom, like something really good model of the protein. Let's say call D01, doesn't matter what D0 is, it's constant. This number is 0.1. So 0.1 squared is 0.001. 1 plus 0.001 is 1.001. 1 divided by 1.001 is 0.995 or something. So it's a number very close to 1. So if the distances are all very small, you're adding up a lot of numbers, 1 plus something a little bit bigger than 1, but not by much. So you're adding up a lot of 0.95s, 0.99. They're all very small. Take the average of those. The max alignments out here means, if I do the alignment 10 different ways, it's the best of those alignments that gave me the best score of this function, this sum. Clear? And so it gives you a number between 0 and 1, because 1 divided by 1 plus some positive number is always a number that is either, if dj is 0, it's equal to 1, but if dj is not 0, it's a number smaller than 1. And you're taking the average over those numbers. So it's always a number between 0 and 1. What is d0? The problem is if you use this kind of a scoring, if you use d0 as some constant, like 1.0, and you evaluate this, if you do a small protein, you get a very different answer. You get different answers for small proteins than for big proteins for the following reason. So what this team did is they took two proteins in the PDB that are not at all related. They're not the same structure. One might be alpha helical, one might be beta sheet. And they aligned the structures with whatever the alignment program was. And then they measured all of these DJ distances. And you get some number. You don't get a very good score. Then you do a bigger protein, 45 amino acids, 50, 100, 1,000, The problem is in the really small proteins, none of the distances can be very big, because if you've overlapped the proteins at all, and the thing is, 15 angstroms in radius, none of the distances can be more than, 10 or 15 angstroms. Most of them will be smaller than that. So you're adding up a lot of small distances, even though the proteins are completely not related. You take a 500 residue protein that is 40 angstroms across and align those, two unrelated proteins, you get a whole lot of very big distances. Again, proteins aren't related, the distances aren't meaningless, but they can all be pretty big. And so you get a much smaller number when you do 1 plus 1 over dj. You're adding up put a lot of big numbers down here. 1 over a big number is 0.1, is a small fraction. And you're averaging over that. So this is what happens. If D0 is a constant across your whole set, so they did this sort of benchmark. They evaluated this expression for constant value of D0. For small proteins, they got scores like 0.6 or something, 0.7, 40 amino acids down here. And they got this basically linear, this is the log scale and the length, but they got this linear correlation with the length of the protein. So huge proteins, 1,000 amino acids, they got, 0.01 or 0.02 or something like that. So they wanted to correct for this, a kind of a size correction. And it turned out if you use something that's related to the cube root of the length of you can kind of think of it as the volume, like a radius from the volume. This is just a fit function. These numbers come from fitting data. To make sure that instead of getting all different scores depending on the length of the protein, I got the same score for the length of the protein. and something kind of small like 0.12. So this D0 is based on the length of the target. It's roughly the cube root. So for a 1,000 amino acid protein, it is something like 10. For a 50 amino acid protein, it is something like 1.1 or something like that. And so it sort of looks like this. Here's D0, here's this cube root function. So it becomes negative for anything that is shorter than 17 amino acids, I think, or 18 amino acids. But here it is graphed. And, so I know it's hard to read. At 200, it's about 5. At 400, it's about 6. At 800, it's about 8. And at 1,000, it's close to 10. So again, roughly the cube root. And so that just scales for bigger proteins. This graph over here is just to show you what 1 over 1 plus some number equals. When the DJ is really small, you get a value towards 1, and then as DJ gets bigger, it goes to 0. But that's just a graph of this. So that's the TM score for comparing two proteins that are exactly the same sequence and the same length. I can do it for complexes. This is sort of like RMSD, where you can align on one thing and align on one part of the structure and score some other part of the structure. Where is it in relation to, So here is a crystal structure of 2 kinase domains, ERK2 and RISC1, heterodimer. If I align on, make sure I do this right. I align on ERK2, which is the magenta protein, in each of these 4 pictures. So here's the crystal structure. If I shift the other protein, RISC1, I shift all the atoms by 1 angstrom, just manually, in PyMOL, just move it. I can evaluate this whole formula, this 1 over 1 plus DJ, again, 1 angstrom, D0, whatever that should be. Take the average, do it multiple times. If I do that, I'm going to call this an interaction or interface template modeling, ipTM, because later we're going to do ipTM. If I move them 3 angstroms, so if I have 1 angstrom, 0.95, move them by 3 angstroms, I get 0.7. If I use a very poor model where even the structure of RISC1 is very poor, it's an AlphaFold model that is not very good, the ITM is 0.5. So again, we're aligning on one protein and we're scoring, we're evaluating all these distances and taking the sum, taking the average, doing it multiple times, and so we're evaluating over, given the position of one partner, we're evaluating the other partner. And that's how ipTM is going to work when we get to it. There is something called DockQ, which was developed to assess. There was a competition called CAPRI, which is related to CASP, which was the structure prediction of complexes of proteins and how good any of the docking programs were for that. So on the left-hand side is 1005 again. It's aurora kinase with TPX2, small protein that activates it, disordered, but binds ordered. Here's the AlphaFold model, which is very good. Everything is really in the right place. How good is this model relative to this crystal structure? And so DockQ is a measure of that. It is based on 3 different numbers. The first one is called FNAT. It's the fraction of the native contacts, meaning if I look at all the residue pairs between the kinase and TPX2, make a list of those pairs that are within some distance of each other, that's a list of contacts. Okay, now I look at the model, what fraction of those contacts did I also get within that distance, that I got a good distance for? So that's the fraction of native contacts that are in the model. The second one is called IRMS, and it's the interface RMSD. Again, it's like RMSD, we already covered that. So I can take all of the atoms and residues that are in contact in the crystal structure. I can make that list, and I can align I can make a list of all of the contacting residues, and I can do the alignment now, not based on the whole structure, but just on that list of pairs. So I'm trying to optimize, residue 1 to residue 1, 2 to 2, 3 to 3, but just of that list of pairs that are in the interface, not the whole structure. And so that's going to tell you, how good are the atom positions of the interface residues. That's called IRMS. And the last one is LRMS, which is an RMSD. It's very much like this one. I align on one of the proteins, say the kinase domain, and then I just calculate the RMSD over the other protein. Again, so it's aligning one part of the structure and scoring, evaluating the position, the model of the other one. And DockQ is just 1/3 of a function of those. So, if IRMS is really big, this number goes down. If LRMS is really big, this number goes down. If the fraction of native contacts is low, the number goes down. So you want DockQ is, close to 1 is a perfect model. 0 is a terrible model. Anything over 0.25, 0.23 is kind of plausible if it's sort of the right, the two proteins have the right relationship to each other. It's a non-zero score. Higher is better. So that's called DockQ. So each of these different measures that we've talked about have been used in the papers, the structure prediction papers like AlphaFold 3. So this graph comes from the AlphaFold 3 paper. And just at the top is just some pictures of some fancy structure predictions that they did. But each of these graphs covers a different kind of target for structure prediction. The first one is about predicting the structures of ligands. Where is the small molecule ligand from the post-buster set from Charlotte Dean's lab. Each of these bars is the percentage of, if you calculate the RMSD of the atoms of the ligand, and if you align on the protein structure and you calculate the RMSD of the ligand atoms, so you align on the residues around the pocket, around where in the crystal structure where the ligand is bound. So you align on those, and you just calculate the RMSD of the ligand. And so what they're claiming here is, AlphaFold 3 gets almost 80% of the pockets have a RMSD better than 2 angstroms. Autodoc Vina is more like 50%, Rosetta Fold a little bit less than that. So they're using RMSD that we've already talked about. The next one for nucleic acids, they're using this LDDT. So this is this local interaction score. You measure all the distances within a sphere, calculate the differences between model and crystal structure. So this is, I think the axis is actually how correct is the actual, it's the distribution of the interface LDDTs for a whole bunch of nucleic acid protein structures. So they're claiming to do better than Rosetta Fold that does nucleic acids. The blue bars are AlphaFold, and the darker gray ones are Rosetta. For covalent modifications like phosphorylation, again, it's that pocket-aligned RMSD. It's looking at where is the phosphate. And then finally, for protein-protein interactions, they're using DockQ, which we just described all of that. That's fancy FNAT, RMS, 2 RMSs. The advantage of AlphaFold 3 now being available to use on our own computers is that we can benchmark it better or on independent sets compared to what the authors of the program did originally. That's good. That's good science. It's really independent assessment of how well things behave. Turns out not as well as they think. So Peter Skriniar in Torsten Svede's lab in Basel did this independent benchmark. And on the x-axis here is they did a whole bunch of ligands bound to protein targets. It's the similarity to any structure that was in the training data. So prior to September 2021. And the success rate, they're saying a docking is successful if the RMSD is less than 2 and the LDDT around the protein ligand binding site is better than 0.8. So it's actually pretty stringent kind of requirement. But it's the protein ligand interface. So again, it's using 2 scores we've already talked about. Well, it turns out for AlphaFold 3, if the target is not related, either the target and the pocket or the ligand itself is not related to anything that is in the training data, they're only getting about 25% of those right. And the other programs are a little bit worse. They Protein X, Chai1, Boltz1, and the best one. If you're very similar to the target, like it's a kinase with a variant of ibrutinib or something like that, you can get 90% of those right, and it's kind of almost linear in between. So you can't take the original publication for an assessment of the accuracy. We need to do this sort of independent thing. I'm not going to go over this graph. This is the model valid? Is the geometry of the ligand good? Are there any bad steric contacts? And it turns out if you put more, it's obvious, if you put more restrictions on the quality of the model being successful, you get an even lower percentage of correct models or successful model. So looking at the geometry of the ligand is important. I won't go through the graph. This was a more recent benchmark of just protein-protein interactions, and it used that DockQ store. Greater than 0.23, the people that run Capri is acceptable. 0.49 medium quality, 0.8 higher quality. This is the percent incorrect for AlphaFold with a template, AlphaFold 2 with no template, and AlphaFold 3. AlphaFold 3 does a little bit better. In other benchmarks, AlphaFold 3 is about this. It's pretty close. AlphaFold 3 and AlphaFold 2 for protein-protein interactions are about the same. So we tend to use AlphaFold 2, but So this is again another independent benchmark using DockQ. Okay, so now to AlphaFold 2 and 3, you take a query sequence, you give it to the program, you search UniPro for all sorts of sequences that are related to it or some other sequence database, and you make a multiple sequence alignment. You could use different programs for that and maybe get different results for that, and different versions of it use different programs for it. You can search the PDB for something that's related to your target, or not. So and find templates, related proteins that are related to your target. If you have more than one target, like it's a protein complex, you would get two different. You might get a crystal structure that has both of them, or crystal structures that just have one of them. AlphaFold 2 multimer and AlphaFold 3 do not use, if you have a template that has both proteins in it or proteins related to both of your proteins, it does not use the interaction between them as features in the model. In other words, it only uses each chain separately to inform the model of each chain. It doesn't use any information in the templates of the relationship between the two chains, exactly how they're docked together. So if you give it a template, it is not using any information of the complex. It's only using each chain separately. Important feature to them. All that information goes into the network. Remember, there are weights, there are sigmas. You get output. There are 5 different sets of weights for AlphaFold 2. We talked about that. You can run multiple times with multiple different random seeds. So start the whole process over with different seeds. There is a random number that is used to begin these calculations. So we typically run it 10 times and we get 50 models, not just once in 5 models. And you get an output structure and you get these scores. pLDDT is for individual residues. pAE is pairs of residues. pTM is the whole structure, and ipTM is for every pair of proteins. So we're going to go through those now. We already covered the local distance difference test. This is the pLDDT. It's the predicted local distance difference test. It works exactly the same way. You pick a residue, you look at all of the distances in the crystal structure, write down those distances, do the same thing in the model, calculate the distances, and what percentage of them are better than these cutoffs. What the network has been trained to do is to predict what LDDT is. So when they train the network, they've got an experimental structure and the sequence. They predict the structure. Now they have a model and they have the predicted structure. Well, for every residue in the crystal structure, they can measure the pLDDT of the model. Sorry, they can measure the LDDT of the model. That gives them a number. They have a part of the network that predicts that number. So they have trained the network to say, well, given the information we had, the sequence, multiple sequence alignment, the template, if there is one, I'll train the network to produce the LDDT, which they can measure. So that, again, that's part of the Y. That's part of the output, predicted output, as well as part of the experimental output. And they can compare actual LDDT with predicted LDDT and optimize the weights to get the best, to get better agreement.
Does that mean that the more people use AlphaFold and the more the network matures, that the pLDDT becomes more accurate?
No, because the network has not changed at all since publication. They don't retrain. It's not like telling ChatGPT something and worrying about whether it's going to tell somebody else. We just call it. So on the left-hand side here is I forget what the protein is, where I've colored the 2 folded domains, magenta and orange. All of the gray stuff is stuff that has a pLDDT worse than 50. And typically the disordered regions all have pLDDT less than 50. If you look at a PDB file, it's in the B factor field, the temperature factor field inside the coordinates. So in PyMOL, I'm sure you can do the same thing in Kimmerer, you can color by that B factor field. And so 100 is, a great pLDDT, 0 is terrible. It's done in percentage. So on the right-hand side, it's the same protein, but I've colored it by pLDDT. The folded parts are all blue, and some of them are like green, and the disordered parts are all red. So the disordered parts will have pLDDTs in the range of 20 to 30. The folded parts will be 70, 80, 90. And then sometimes you might get some pieces that are 60, 70. So here's the command in PyMOL, color gray, comma, B less than 50. To do all the residues, you can use the color menu to color by the B factor. That's pLDDT. Here it is for KRAS. These are just the 5 models of KRAS. This is the switch 1 loop and switch 2 loop. Those are the ones that move around. The models are slightly different from each other. And here's the pLDDT goes down to 40 in the switch 1 and 50 in switch 2. And the folded parts are all sort of 80, 90. So that's just what it looks like for a single protein. Okay, now the cream of the crop here is PAE. A lot of things depend on PAE, so we'll spend some time on this. Predicted aligned error is obviously going to be based on something called aligned error. And so the aligned error is the concept that we have to get here. So the aligned error, and it's related to things that we've already done, so hopefully it'll be intuitive enough. We have a experimental structure of a protein and a model of a protein. So over here, this is, again, 1005, the Aurora kinase. I've aligned the two structures based on just the N, C alpha, and C atom of one residue, this magenta residue. It's the aspartic acid of the HRD motif. But you can see the rest of the structure kind of follows, even though I'm not aligning the whole structure, I'm just aligning on 3 atoms and 1 amino acid. But if I do that, I can then look at another residue. So this is, in Aurora, this is 185 or something like that. Here's residue, I don't know, 288 in the PDB. And in the model, it's way over here. So there's a distance between those. So, it didn't, after aligning on this, the model amino acid is, quite some distance away from the PDB one. That's the aligned error. So given for those two residues, so given I've aligned on one residue and I've scored the other one. We've talked multiple times previously today, I said, align on some part of the structure, score another part. This is aligning on this residue and scoring the position of this part. How far away is it? If I've aligned on that one, how far away is the model residue and the PDB residue of one particular residue? On the right-hand side is what a graph looks like for a protein called BTK, which is a kinase. And so the aligned residues, the sequence goes from 1 to 659. The aligned residues are on the y-axis, and the scored residues are on the x-axis. That means right here, if I align on residue 100, and I score all the residues from 0 to 150, well, let's do 150. If I start right there, residue 150, and I score all of the residues in up to here, they've all got really good scores. They've got an aligned error. This is the predicted aligned error. So the network has been trained to predict these aligned errors. So that's why it's now PAE. So if I align on one residue and I score all the other ones, well, from within the same domain, that's this blue thing up here, it's pH domain. Everything is dark green. The values are 2 to 3, 4 angstroms. But outside of that domain, this blue domain is not related, its position is not related to the position of the other domains in the protein. So it scores rather poorly with everything else. But as you can see, it's a square. These are all of the pH, it's called the pH-TH domain, all related to each other. That square is that domain. Everything has pretty good PAEs with everything else inside the same folded domain. Here's the SH3 domain, it's this dark square. Here's the SH2 domain. But SH2 to SH3, there is kind of, darker green, not as dark as within a domain, but decently dark. The relationship between SH3 and SH2. So SH3 is this right here, SH2 is this. These two domains and these kinds of kinases are always kind of the same orientation with respect to each other. And AlphaFold knows that. It knows that the two domains have a relationship to each other. That's this green stuff that is kind of off the diagonal. The kinase domain is here, big square for the kinase domain, and the position of the kinase and the SH3 domain in particular are kind of strongly related to each other, and the PAEs are pretty good. So you see these, drawings, and you get very clearly the domain structure of the protein, or the big squares along the diagonal. And all the off diagonal stuff, besides those dark green squares, the stuff off the diagonal is the relationship between the domains. So is that all clear?
And the plot is like almost symmetric, but it's not.
Yes, it's pseudo-symmetric. It is not completely symmetric. Just mathematically, numerically, it's never going to be exactly symmetric. It's never really clear whether, which rectangle I should look at to there are different ways of thinking of it. Okay, so this is going to get a little mathy. And if you don't get it when I do the equations the first time, that's totally understandable. Looking at equations on a screen is not, nobody learns math that way. Well, I suppose in high school you do, but it may not be immediately clear as I'm saying it today, but you're going to get the slides and you'll be able to think about it a little bit more. So we'll go as follows. We already talked about the TM score. it's this 1/1 plus this thing, the DJ. So here's the aligned proteins. The DJs are just the distances between the same amino acids in the model in crystal. And it's the maximum over any set of alignments. It turns out the PAE is exactly the same as the DJ. So remember, the PAE is aligning on one residue, aligning on some part of the structure, and just measuring the position of some other part of the structure, some distance between the other part of the structure. That's exactly what this is. This is an alignment of the two proteins and just measuring the distances between model and PDB. So from this expression, from what AlphaFold has been trained, it's been trained to calculate PAE, I can calculate the TM score for the predicted model just by putting the PAEs down here. And every residue so every residue in the protein, remember, you align on one residue and you score all the others. So I'm aligning on each residue here and I'm scoring all the others. You can think of this as 659 separate alignments. In other words, I've aligned on residue 1, 2, 3, 4, 5, all the way up to 659. And I've measured I've predicted the position or the error in the position of all of the other 658 residues. And so this is like having 659 separate alignments of the model to the PDB structure. And so that's why this maximum over all the residues. So each one of these is an alignment. And each one of these is scoring the whole protein given that alignment. So that's why it's PAE, I, residue J, squared, and then the D0. So again, that's aligned residue, scored residue. This max I is aligned residue. The J here, J equals 1 to L, is the scored residue, and I just take the average. I know that's a little complicated.
Question. So using PTM together with PA as a filtering score doesn't make sense because they are related to each other.
Sorry, say it again.
Using both score, PTM and PA as a filtering score doesn't make sense because they are connected.
Yes, they are related to each other. The PAE is only for any pair of residues. You can take an average over all the interface contacts. You can take selections of PAEs and calculate averages or minimum values over them. But you're right, the PAEs are what is used to calculate ipTM and ipTM. The ipTM and ipTM are kind of summary statistics over the whole PAE matrix. And so they're valuable because they're just a single number between 0 and 1 as opposed to a whole matrix. But it is always, if you do a model in AlphaFold 2 or 3, look at this matrix and figure out what it's telling you about the domain structure of each of one protein or two proteins and how those domains might interact both intramolecularly and intermolecularly. So that's so the PTM is just predict the TM score using the same mathematics as go with the TM score. Predict that TM score for a whole protein or a whole complex of proteins. So it's just done over all the residues, aligning all the residues and scoring all the residues, throughout and getting the maximum value and getting that PTM. You can get the PAE for protein complexes. This is a complex of the protein RAF1 and KSR1. RAF1 has a RAS binding domain, a cysteine-rich domain, and a kinase domain. So that's this magenta and this sort of pink stuff here. KSR1 has a coiled SAM domain in green, another cysteine-rich domain in yellow, and the kinase domain in blue. So there's the green, there's the yellow, there's the blue. And there's lots of disorder in both proteins. Here's what the AlphaFold PAE matrix looks like for this complex. So along the diagonal is, amino acids against themselves. So here's, the upper left box is RAF1 versus itself. So it's square because it's, same number of amino acids, about 600 by 600. The kinase domain is here. It's in bright blue because everything within the kinase domain is pretty well predicted to everything else except the activation loop. RBD to RBD and CRD to CRD, also blue, and also, some decent relationship with each other. The scores, this is a red-blue, instead of green, white, this is a red-blue thing, but it's the same idea. Blue is good, blue is low, white is medium, and red is bad. Same thing for the KSR1. Here's the CC-SAM domain against itself, CRD against itself, kinase domain itself. But the off-diagonal rectangles are the relationship of one protein to the other. So down here, I'm aligning on residues of KSR1 and I'm scoring here I'm scoring residues of RAF1. So this is KSR1, this is RAF1. So the kinase domain, the kind of interaction of the blue kinase domain and the magenta kinase domain, that all scores really well. There's good PAEs between those two domains, one in each of the two proteins. Same thing going the other way. If I align on RAF1 residues and I score KSR1, the only part that's blue is the kinase domain. So the two proteins don't care about the CC-SAM CRD domain, all the disorder, the RBD CRD domain, are not part of the evolved interface, and they're not scored very well by AlphaFold. So you always want to look at this complex and look at the off-diagonal rectangles, so sort of lower left and upper right, to see if there's anything blue. If there's nothing blue, AlphaFold is not telling you anything about how your proteins interact. You need to see some blue in order to believe anything.
Explain again why the off-diagonal patterns, the matrices, look slightly different, whether you're aligning with RAF and scoring with KSR1 and.
Yeah, I don't ultimately know the exact reason for it. It's kind of a numerical thing that it's predicting that it's an asymmetric score. You're aligning on one thing, scoring the other. And it's been trained to, well, if I align on one thing and score the other one, this is the number this is what I think the error might be in that distance. And the model just isn't completely symmetric. If I look at residue 50 in one protein and I score residue 75 in the other one, there's a certain, what's the error in residue 75? Whereas the other way, it's aligning on 75 in this one and scoring residue 50. Those are not the same thing, right? One is the error of position 75. The other one is the error in the position of 50. And so they're not identical. You're not calculating identical things. That's why it's asymmetric, kind of by design. Aligning on one thing, scoring another. And so if you do it, align on this one, score this one, you get one number. If you align on this one, score that one, you get a different number. Usually closely related. They're not going to be, they shouldn't be wildly different. I don't know if anybody spent a lot of time kind of analyzing how asymmetric they are and when are they asymmetric and does that tell you anything? I don't know of anything like that.
That's a related question.
Yeah.
So from the DA block, let's say you have kinase dimer, then you have a head-to-head or head-to-tail or back-to-back.
Yeah.
The difference between those constellations and what's the constellation of the condition? Can you figure that out from the DA plot if you have different constellations?
A PAE plot is for only one model.
Yes.
And so in that model, you would then look at the coordinates and see, oh, it's back to back. And then another model, you might still see, blue in the PAE in the right region, but they're face to face. The question is, are the scores equally good? Maybe both models are correct. Or one of them, that's what happened in the model and the scores are very poor. So, it's 1-to-1, one PAE matrix to one model.
But is there any way to compare to the matrices? Because in the ultimately, you get an ipTM score, but I just looked at the PA plot for one model. Is there any way to assess the quality of compare the quality of human ones?
You would calculate any of the quality measures on each one separately. We get different numbers. The shape and the form and the exact dimensions of the blue rectangles might be telling you something, and whether they're very strongly predicted. We usually see it. We usually see, do all the PAE matrices, and a whole bunch of them look the same, and then something else happens, and they go away, or they're there, but they're much, they're white, not blue. And then you look at the models and go, oh, all of these look alike, and these look different. And usually you're only getting one correct protein-protein interaction that scores well, and anytime you don't get any good scores, two proteins are all over the place with respect to each other.
So then you rely on the average and models.
You could do that. Yeah, you can take the top set of models and take averages. So, okay, so now we get into ipTM, which is the interface predicted TM score, template modeling score. So the I is for interface. It is not about the interface. The interface has nothing to do with it. Total misnomer. But again, it's predicted TM score, PTM. So here's our same pair of proteins, KSR1 and RAF1. Here's the domain diagrams for the two proteins. If I calculate, so ipTM, which I haven't told you how it works, but if I told you how it works, the number we get is 0.38. If I isolate just the two kinase domains, I get 0.82. So in other words, I've given AlphaFold just the 300 residue sequences of the two kinase domains, 300 each, and I get 0.82. But the model is the same. Like the predicted, the interaction of magenta and blue is the same here as it is here. But the score here is, not great. They're 0.38, and here it's 0.82. Why? And is there anything we can do about it? So AlphaFold calculates something called ipTM. We've already talked about TM and then PTM, and this is ipTM. Remember, it's asymmetric. You're aligning on one thing and scoring another. So I can align on the residues in chain A, and I can score the positions, the PAE values for all of the residues in chain B. That gives me an idea of whether the orientation position of B relative to A, if I fix on A, align on A, where is B? And so that's what the ipTM is. It means I'm going to look at every residue in chain A, so max all the I residues in chain A, and I'm going to evaluate the mean value of this expression for all the residues in chain B. And again, that's 1 over 1 plus PAE ij squared divided by something called D0 squared. And remember, the PAEs are really big. This is 1 over a big number. It's 0.1. If the PAEs are all 0, like totally well predicted structure, that's 1 over 1 plus 0. It's a whole lot of 1s. Take a mean of a whole lot of 1s, I get 1. Take the maximum, no matter what, I'm going to get 1, something close to 1. So it's asymmetric. I can align on one chain and score the other one. That D0 in AlphaFold is calculated based on the length of both sequences combined, which is kind of a strange choice, actually, because you would think the D0 should just be related to the scored chain, the size of the scored chain, not the size. You shouldn't have anything to do with where you do the alignment. It's only on the parts that you score. But it isn't. They do the cube root of the sum of the length of these two proteins. So for this one, there's 1,500 residues. It's a cube root of 1,500, which is something like 12 multiplied by all these numbers. And so the final ipTM that you get is actually just the maximum of the two asymmetric scores. If I align on chain A and score chain B, that's one number. If I align on chain B and I score chain A, that's another number. Which number is bigger? That's the ipTM. And that maximum comes from all the sets of alignments. It's that original TM score. Align it in different ways. Take the maximum of over all the alignments of the mathematical expression. So that's ipTM, and it's based on the PAE. In the program itself, AlphaFold calculates a probability distribution over PAE. It's a little bit more complicated, but it basically is the PAE. So most things depend on the PAE.
The two peptides?
What?
What if you have more than 2 peptides?
This is a complex of 2 proteins.
What if you have like 3 proteins? How is the ipTM calculated?
Oh, what if I have more than 2 proteins? AlphaFold 2 will calculate one ipTM for the whole thing. So it's basically one residue and one protein scoring all the residues in all the other chains. AlphaFold 3 said, well, we can give an ipTM for every pair of proteins. So it gives you one for every pair. It doesn't give you the asymmetric values. It gives you just one value, symmetric. But you get one for every pair. And very frequently you get, the 2 proteins make a really nice complex, but the third protein is really uncertain with regard to either one of them. And you need to pay attention to all the pairwise values, not just the overall. AlphaFold 3 will also give you one value for the entire complex. That number is pretty useless when there's more than 2 proteins. So this mathematical formulation explains why, if there's lots of disorder and extra domains in both proteins, ipTM is kind of artificially low compared to really what it really should be, given the look of the PAE matrix. And the reason is this: if I align on a residue in chain A, and I'm scoring all the residues in chain B, I'm evaluating that 1 over 1 plus DJ stuff. I'm evaluating over a lot of disorder, 600 residues of disorder and 300 residues of order. And so I'm going to get a lot of 0.9s for the ordered part and a lot of 0.1s for the disordered part. Same thing if I align on a residue in chain B and I score only chain A, half of it is disorder and half of it is order, 300 and 300 residues. And so the ipTM comes out about 0.4. It's probably aligning on this residue here, scoring half disorder, get a lot of 0.1s, scoring completely ordered, getting a lot of 0.8s, 300 of those, 300 of those, and you get 0.41. Right. So in the AlphaFold multimer paper, they use DockQ to measure the accuracy of structures. And they say, well, how accurate is the structure if the I interface PTM is between, now it's multiplied by 100, between 0 and 10? Yeah, the DockQ scores are really terrible. You don't really get decent DockQ scores until you get an ipTM of about 65 before you start, getting kind of 40% of the models are pretty good, 50% of the models are pretty good. Not until ipTM is about 0.7 or 70. Again, this is in there, this is Alpha Multimer 2 in March. I think this is, I forget whose benchmark this is, but anyway, ipTM tells you something about the accuracy of the interaction between the two proteins, which is measured by DockQ. Our solution to this, once I finally kind of dove into the math and figured out, oh, that's how ipTM works. I had to look at the code to figure out exactly what it was doing. In AlphaFold 2 and then the AlphaFold 3 supplement gave really clear equations on this. There's a way to fix it. There are several ways to fix it, but there's one way we do it. It's called ipSAE. It stands for Interaction Prediction Score from Aligned Errors. Notice this doesn't say interface. ipTM is evaluated over the whole structure. So if I line on chain A, it's evaluated over all of chain B. It has nothing to do with the interface. It is not calculated on the interface. That's got to be very clear. It turns out a lot of the residues in the interface have good PAEs, and so the scores, those residues contribute. But it's over the whole domain and the whole domain of the two proteins. That's what that blue square is. So the way to solve it is, what if I only take the part of this off-diagonal rectangle here, and I put a cutoff of the PAE? I'm only going to look at the parts of that rectangle where the PAE is better than 10 angstroms, which is pretty generous. That's like several amino acids. Like it's not a very stringent cutoff, 10 angstroms. But if I evaluate the ipTM expression just over those residues, so that's what we do. We just take, okay, this internal expression here. I'm going to align on every residue of A, take the maximum value over residues in chain A. I'm going to score the residues in chain B, but I'm only going to score the residues where the PAE IJ is better than the cutoff, let's say 10 angstroms. And that's the same 1 over 1 plus PAE squared. And I'm going to evaluate only over that part. So for the kinase-kinase interaction, I'm only evaluating this over the kinase residues of the partner protein, and aligning for all the residues over here. The D0 is really important here, actually. This scaling thing for the size of the system is actually very important. What we do in ipSAE is, oh, well, I'm only scoring a certain set of residues over here, all the ones that have better than 10 angstrom. I'm going to calculate the D0, which is a cube root of the length of the protein, over that length, over the number of residues that are scored. And so it makes the two things very closely related to each other. The sum, the mean, is taken over the residues that are better than the cutoff. The D0 is calculated on the residues that are better than the cutoff. If the length is greater than 27, then we just set it to a minimum value of 1. That just turned out to be more numerically stable. So this is called ipSAE, and it's because it's a Latin word. I'll explain that. But the reason it works kind of more visually is this. If I align on a residue in chain A and I score chain B, I'm only scoring the blue stuff. So I'm only scoring this bit over here. Again, align on a residue in chain B and score chain A, I'm only calculating a TM-like score on the blue part. And so ipSAE is 0.75 for the full-length proteins and 0.75 for the complex of just the two domains. So it fixes the problems of all the extra disorder or extra domains that are not interacting in the two proteins. You could solve this in different ways that wouldn't work. You could look at all the interface residues, but there's a lot of interface interactions between these domains that don't, and they look different in every model. There's a lot of interface contacts between the two proteins down here that score really poorly, in here. AlphaFold doesn't think anything of those, and in every model they would be different. It only thinks there's good PAEs between the kinase domain. So using interface as a way to separate good from bad is not helpful here. Using pLDDT is not that useful. pLDDT, the gray part, the disordered part here, is all worse than 50. These things have good pLDDTs because these are nice folded domains. The pLDDTs tell you a little bit about whether there's an interface between two, but there are good pLDDTs throughout these folded domains. pLDDTs don't tell you whether the protein-protein interaction is good. Let's get that. It's a little hard to explain. So these are calculations over RAF1 and KSR1. The RAF1, just the kinase domain against KSR1, RAF1 versus KSR1 kinase domain, and the kinase domain versus kinase domain. Number of residues involved, here's that D0. So my D0 is based on the number of residues that pass PAE. It's always about 6.2. D0 as the cube root of the length of the two proteins goes from 12.6 down to 8.8. So the length of the proteins matters. If you start adding lots of extra disorder to one of the proteins, the ipTM score actually goes up because you're scaling these D0 by a bigger number. So it's kind of another flaw in the ipTM formulation. So I think our formulation of this is pretty good. It works pretty well. There are other ways to do this problem. But let's just see how it works for complexes. If I take 40 structures from the PDB that are not in the training data, not very closely related to anything in the training data. So 40 different complexes. I take the full length uniprot sequences, not just what the author crystal structure might be just one domain and one domain from very long proteins, but that's all they did to crystallize it. I'm using the full length uniprot sequence, which might be 1,000 amino acids, including lots of extra stuff that doesn't really interact. I then make I made a set of 70 that are non-interacting ones. They're actually just taking all the human pairs in this and just kind of scrambling the pairs. So these are proteins that shouldn't interact with each other, but they're the same sequences that are in the interactors. They're just scrambled between different PDB entries, different structures. So anything that is going to be a good classifier, whether this is a true interaction or a false interaction, is going to score true complexes really highly and poor complexes or false complexes, fake ones, really poorly. This is what ipTM does. The blue curve is all the true complexes. Yeah, the peak is at about 0.9, but there are lots of true complexes that are down here, which isn't great. And the false complexes, yeah, the majority of them are around 0.2, but there's also some false complexes that are scoring 0.4 up to 0.6. The ipSAE score run at 10 angstroms. The true ones are all up here. They're basically almost all better than 0.4. There's a few down here that AlphaFold didn't detect an interaction. All the fake complexes are actually all almost exactly 0. They're either 0 or just slightly bigger than 0. And so there's very little overlap between these two distributions. So ipSAE is a better number for detecting true interactions from unlikely interactions. It's not perfect. AlphaFold might not detect an interaction between two proteins that interact, doesn't score any good PAEs, and yet the two proteins interact. You can see them in a crystal structure perhaps. And it's just, for whatever reason, it's a false negative. AlphaFold itself didn't detect an interaction between the two proteins. No scoring system in the world could turn that into a good score if AlphaFold doesn't detect, has no good PAE. If all the PAEs between the two proteins are 30 angstroms, it just means that AlphaFold did not detect anything between them. None of these scores will fix that problem. If we change the D0, we get, here's the D0 based on the full length chains. Again, I think that's a bad idea doing them on the full length chain, but that's what AlphaFold does. It's worse. There's more overlap between the two curves. If you do it the way we do it with only the number of residues in the PAE, better than the PAE cutoff, there's very little overlap between the two curves. There are other solutions to this problem. And I've already talked about using pLDDT to restrict it. That doesn't really work because you have good pLDDTs in the folded domains that are not part of the binding interface. You can look at just the contact residues. Again, I said there are incidental contacts between two proteins that are scored poorly and have nothing to do with the interaction. So p.q is kind of an average over the interface PLD. Overall, the pLDDTs of all the residues in the interface between the two proteins. Again, is just really not a good way to do it. p.q2 is a variant of that. Instead, they use the PAEs of all of the residues in the interfaces. Again, there's a lot of contacts between two proteins, multi-domain proteins, that'll score really poorly on the PAE chart and shouldn't be part of the evaluation of how the two proteins interact together. So p.q2 will fail for very long proteins with extra domains. A more recent version, active pTM, looks at called the distogram. It's the prediction of the distances between two amino acids, and so they get all of the residues that AlphaFold predicts a short distance between a residue in one protein and a residue in the other one, and then they evaluate the pTM-like expression over that set of residues. That's a reasonably good idea. The problem is they set D0 to the sum of the ipTM version of D0, the length of the two chains combined, take the cube root, as opposed to just a set of residues that are getting scored. And that's a mistake. It gives you values that are highly inflated compared to what they really should be. There's also the LIS and ILIS and CLIS, local interaction score. it's another smart way of doing it. It takes the PAE matrix and says, well, I'm just going to look at the parts that are better than 12. So we did 10, they're doing 12. And instead of using this 1 over 1 plus, formulation, that function, they're just going to convert the PAEs to a number between 0 and 1 as a linear transformation. So if the PAE is 12, that's a 0. If the PAE is 0, that's a 1. So really short PAEs are scored near 1. Really long PAEs, close to 12, are scored just a little bit above 0. Anything worse than, and it's just an average over those. It is an intelligent way of doing it, and LIS and ipSAE are kind of rival scores, but they're very closely related to each other in a way. And I'm not sure whether, when, and where there's an advantage to one over the other, to be fair. I'm not going to go through it, but this chart is exactly how they do the calculate the LIS. And then they have a version of it where they just look at the contacts that are better than PAEs better than some number. That's called CLIS. And then they combine those two to get ILIS. But here's the math on that. I don't want to spend more time on it. I can if somebody has a question. So if we do that same set of that benchmark on ipSAE and LIS, p.q, p.q2, p.q and p.q2, there's a lot of overlap between the true interactors and the false ones. LIS, there's less, there's that, and ipSAE, I think there's somewhat less in ipSAE than there is in LIS, but it's not a big difference. So these two scores I think are pretty good. These scores are, I think, actually pretty poor most of the time. The code is on GitHub. It is just a Python script. You give it the JSON file from AlphaFold 3 or AlphaFold 2 output, the big JSON files that have all of the PAE matrix data in it. Then you give it the PDB file on the mmsif file. You give it a PAE cutoff. We usually use 10, sometimes 15. There's a distance cutoff, which is really, I'm going to say, all the residues that pass the PAE cutoff, how many of them are within some distance between the two proteins? It just gives you a number. This is the output. Remember, it's an asymmetric score. You can align on chain A, score chain B, align on chain B, score chain A. So that's, I know it's hard to see, but this says asym. So this is aligning on chain A, scoring B, aligning on B, scoring A. Here is the ipSAE score here, this 6-digit number. And then, the numbers are a little bit different in this case. This is for a 3-protein complex, KSR1, RAF1, and MEK. And so you get A to B and A to C and B to C. You get 3 sets of numbers for the 3 different pairs. It calculates a lot of the other scores as well. So you can compare the LIS score and the p.q score. It's not actually in this chart, but it's in the current version of the code. And there's a PyMOL script that outputs that says you run it, and there's a little alias says color AB, and it colors all the residues in each of the 2 chains that have good PAEs with respect to the other chain. So for RAF1, KSR1, chain A and B, you see that interaction. For KSR1 and MEK1, there's a good interaction. So there's a good score, ipSAE score between chain B and chain C, which you see right here. And so you can call, and you can apply it to a whole set of models, and it will tell you what parts of one chain are interacting with what parts of another. It's very useful. It comes from this Latin expression, res ipsae loquuntur, which means it's a pronoun, which I like pronouns. People should use their pronouns that they wish and be addressed by them. Ipsae means themselves. So the things themselves speak. It's used in legal circles in the singular, res ipsa loquitur. Means the thing, if you see a car accident, then somebody is at fault, something went wrong. So the fact that you see something, the thing itself speaks to you and says, somebody's at fault, something went wrong, we need to find it. So this is just a plural of that. It's from a lined error. I wanted an AE. What language has AE in it a lot? Latin. Try to finish up the applications here. And then we'll be pretty close to the next section we're going to do with some kind of the demonstrations. So how is ipSAE used? And the LIS score can be used in the same way. PPI screens, where I do one protein against 500 different proteins that might interact with it. It's good because you don't have to worry about breaking things down into all of the domains that individually interact with between the 2 proteins. You can just do the full-length pair of proteins, get a good score, and then look at the PyMOL or look at the PAE chart and figure out, well, what domain of one protein interacted with what domain of the other protein, and then investigate it further. We used it for binding specificity between E2s and E3s. Humans have 37 E2 proteins and 400 E3s. So that's in that preprint. Let me just I'll show you some of the pictures. AlphaFold database is now, so AlphaFold, they did all 200 million UniProt sequences. So the AlphaFold database, you can look up any protein in UniProt, and there is one model. They only built one model with model 1 of AlphaFold from 2021. So they only used model 1.1.1. It's the first of 5. When you run CloudFold, you get 1, 2, 3, 4, 5. AlphaFold databases they only ran it once and only on model 1. That's how they did 200 million of them. So that's why when you can take a look at that model, but you really want to run 50 models of your own because they only ran one model of one with one seed. They are now doing homodimers and heterodimers. So you look up your protein, you can get, is it a homodimer? And if it scores well, it's they upload a homodimer model, and they're now doing heterodimers, your protein against every other protein in the same species. So they benchmarked ipSAE and some of the other scores of true homodimers in purple and monomeric proteins. If they made homodimers, what score did they get in this brown? And all the true ones are up here and all the false ones are down here. You can see the axis here is 49.7. And so it was somewhat better at separating true from false true homodimers from false ones. So when you go on there, it gives you the ipSAE score between the two proteins as well as p.q. I give you LAS, I'm not sure. Let's get that. We're using it intramolecularly. So this is a protein, the NEC1 kinase, has a kinase domain and a novel domain that has no name. There's this little 3 helical bundle down here, which I'm just calling NEC C-terminal domain. So I wrote a very simple, ChatGPT wrote a very simple script to take a model of a single protein, and I give it some residue numbers, like the boundaries, residue 1, residue 258, each of these little helices. This begins on 1175. And it just outputs a PDB file where each segment of that is given a different chain ID. So then I can just run that script is available on the GitHub. Now I can just run it on a single, now I'll get an output PDB file that I can run with the same JSON file. And now it scores, ipSAE score between chain A or domain A against domain B, domain C, and domain D, and you get a score. So you're getting an intramolecular domain-domain interaction score. I don't know that any of the other programs have done this. It's not hard to do, but they could do it. But anyway, this little helical domain is binding to the kinase domain, I think in a way that makes the kinase domain inactive, and the score is 0.45. And as I showed you, the score is really above 0.2, or generally the true ones are above 0.2, and certainly above 0.4, they're usually pretty good scores. So this is an intramolecular ipSAE to do domain-domain interactions inside of a single protein. We're doing these, bio-ID screens and taking we're doing a lot of stuff on kinases. One kinase against all of the proteins that it might interact with. This is just the Cask pseudokinase. And so we're taking all of the individual domains of Cask. We're giving it the entire protein Cask, which has all of these different domains, running different proteins. And it turns out, not surprisingly, the different proteins interact with specific domains. And we're getting good scores, ipSAE scores between the two proteins. And the PyMOL session or the output is telling us, oh, it's this domain of Cask and this domain of the other protein. So not only do you get score saying, yeah, these proteins likely interact, but also you get the structure and what parts of the two structures interact. That's the advantage of this, which you can then use to design mutations, experiments to test things. People are using this a lot for protein design, where you've got as you see in the upper left here, a target protein, and you're using Rosetta fold diffusion or Boltzgen, or there's a whole bunch of them, that will design a little mini protein that will bind to the surface of that particular surface that you're interested in to maybe block the protein-protein interaction. This is a very big field. This is what David Baker got the Nobel Prize for in 2024. The people that do protein design are using ipSAE to determine, well, I did 10,000 designs on the computer. I can't make 10,000 designs in the lab. They use an ipSAE score. Basically, they make the design, predict the structure with AlphaFold 2 or 3, score it with ipSAE or LAS or any of the other scores, and say, okay, if the scores are below some value, I'm going to throw out all of those sequences. I made 10,000, 6,000 of them, or 2/3 of them maybe, score poorly. I have no confidence in them. And it will turn out experimentally like 90% of those would not bind. Some of them would, but very, very few. If the scores are above 0.6, then about half of those designs will bind. So they still need another way of ranking. It's not very good at ranking within that upper set, but it's pretty good at cutting out the vast majority of non-binders. And then you have to do other things to try to figure out which 10 should I make in the lab. So these three preprints are doing that. My colleague, my friend Richard Bayless at the University of Leeds did this for Aurora kinase. Here's the kinase, and here are all the designs they made. And they did crystal structures of some of them. But just here's the ipSAE result. For these three are true binders. The KD and nanomolar is 1, 3, and 22. They all had good ipSAEs above 0.6. These three had, ipSAE well below 0.6. And, they still bound, but they had, 120 molar. This one is an outlier. This one had a good ipSAE, but had a poor KD. So it's sort of decent at picking out, true binders from false ones. Okay, I'm going to give you a 15-minute break now. I have a little bit more on AlphaFold, but then we will jump into, demonstration, kind of looking at some of the websites where you can do some of this stuff. I suspect most people don't want to sit here and try to do something on your computer while I'm doing something up here, but you can. I can, show you how to do that. So let's come back very promptly at 20 of 4.